if you focus on the phenotypes, you'll get 3 out of 4, or should I say the probability of having the offspring showing the dominant phenotype is 3 out of 4, and the probability of the offspring showing the recessive phenotype is 1 out of 4. But if you concentrate on the genotypes, the probability that any offspring will be homozygous dominant is 1 out of 4. The probability that any one offspring will be heterozygous is 2 out of 4. And the probability that any offspring will be homozygous recessive is 1 out of 4. Again, those probabilities are straight off the Punnett square there. And maybe I should take a moment to talk about how the terms we use when talking about these probabilities. In some cases, we can just use fractions. Like I might say, what's the probability of having a little p plant from this monohybrid cross? And so sometimes I might say, well, it's 1 out of 4. Call it like that. Or I might use percentages instead. I might say it's a 25% chance that we'll get a homozygous recessive white flower plant off this. Or I could talk about ratios. I could say for this particular crossing, I expect a 3-to-1 ratio of purple to white. So be ready to see the probabilities expressed in any of those 3 ways. All right, well, let's see. So one of the concepts that came up last week was this. If you see an organism that's showing the recessive phenotype, the recessive physical trait, like the white flower, you know it's a genotype because you only get the recessive phenotype when you have the homozygous recessive genotype, little p. But if you see an organism displaying the dominant phenotype, like purple flowers, you don't know its phenotype exactly. You could get the purple flowers if you're big P, little p, or you could get the purple flowers if you're big P, big P. And so if you did have a purple plant and you were really curious about, well, is it this genotype or is it this genotype? How could you tell? Well, Mendel came up with a concept called a test cross, which is a way of figuring out whether your dominant organism is big P, or big P, little p. And what Mendel said was this. Oh, there's the monohybrid cross, okay. Here we go. So Mendel said, if you're curious, mate it with a homozygous recessive. Now I want, don't peek at your notes. Let's say everybody on this side of the room, scribble on the back of your notes somewhere a Punnett square that shows big P crossed with little p, and let's discuss what sort of results you get. And everybody on this side of the room, do a Punnett square of big P crossed with little p, and tell me what sort of offspring you expect. Anybody have an answer? You got one, Louis? What do you think? Yes. Of what genotype? Okay, and for the offspring, what was their genotype going to be? Big P, little p, yes. There it is. So if the plant you're talking about was homozygous dominant, the purple plant, and you cross it with a homozygous recessive, if you do the Punnett square, all the squares have to be big P, little p of the offspring. So each offspring has a 100% chance of being big P, little p, which is purple. So you should get all dominant phenotype offspring. If the parent plant was big P, little p, what would you expect from the offspring? Someone over here? Half, exactly. Let's see. There we go. Oop, come back here. There we go. No, that's not it. There we go. If the purple parent was big P, little p, and you did this test crossed with the homozygous recessive other parent, notice that half the squares of the Punnett square give you the dominant phenotype, big P, little p, purple plants, and half would give you the recessive phenotype, the white plants, little p genotype. And so here those are spelled out. If you're not sure whether your dominant plant is big A, or big A, little a, cross it with a little a plant. If you get all dominant phenotypes in the offspring, your parent plant was probably big A, but if you get some of the recessive trait showing up in the offspring, then the parent must have been big A, little a, the parent in question. Does that sound all right? Okay, now, up to this point I've been focusing on just one trait in Mendel's crosses, which are the purple flowers. Mendel also looked at another trait, tallness trait. Some plants had the normal amount of tallness, which I'll show here, but he also found some of his peas had a dwarf phenotype, like this. Well, this is a good chance for a review. What letter might Mendel have picked? Oh, I'll tell you, the tall is the dominant trait. He probably would have used T, and capital T for the dominant trait, the tall ones, and little t for the short recessive trait. And as it turns out, the little t is the recessive trait in this one. And let's say he had a plant that was heterozygous for the purple trait and heterozygous for the tallness trait, and he was going to cross it with another plant that was heterozygous for purpleness and heterozygous for the tallness trait. Well, if we want to do a Punnett square of this, remember that to do the Punnett squares, we write the genotypes of all the gametes that can be produced. And here's where things get a little tricky. Here's this parent plant right here, big P, little p, and big T, little t. What sorts of gametes can it produce? Well, remember that each gamete, sperm or each egg, is only going to have one of these flower color genes and only one of these tallness genes. But which one it gets is random, whether half the sperm will get big P allele and half the sperm will get little p allele. And likewise with the tallness alleles, half the sperm will get the big T and half the sperm will get the little t. But each sperm has to get one of the flower colors and one of the tallness alleles, and it's a random 50-50 mixture of which one you're going to get, or I shouldn't say 50-50. All combinations and permutations are equally likely. So it's possible, for example, that a sperm that's being made by this parent will get the big P allele for its flower color and the big T allele for its tallness genes. But it's also equally likely that the sperm could get the big P allele but get the little T allele. And so this is equally likely to make some sperm like that. And it's also equally likely that a sperm could get the little P allele for flower color and the big T allele for tallness. So that's as equally likely as anything else. And it's also equally likely that a sperm can get little p and little t. Like that. So again, all possible combinations come up in exactly equal amounts. You always get one of each gene, so one of these P's and one of these T's, but which one you get is completely random, and so it comes out to be equal numbers. And here's something from the earlier in the lecture from last week on Mendel's rules of genetics. I skipped that part, but this is what this is illustrating. Gametes with all possible combinations of the maternal and paternal genes are made, and all combinations are made with equal frequencies. That's called the law of independent assortment. And if you're trying to figure it out, maybe just do it the way just I did. Take the first allele and draw little arrows from A to big B and big A to little b, and write those, then go with little a to big B and little a to little b. And we'll get all gametes. 25% of the gametes will be this, and 25% of the gametes this, and 25% of the gametes this, and 25% of the gametes that. Okay, so now we can go back to this cross looking at two genes we're talking here. You need a bigger Punnett square. Oh yeah, so there's the gametes this thing could make. Equal numbers of all four of those gamete types. And so you need, each parent's going to make 4 types of gametes. There's from that parent, there's from that parent. And so notice things have gotten a little more complicated now. You need a 4x4 Punnett square. And there are the possible genotypes of the offspring. So it does look kind of complicated, but it might maybe not quite be as complicated as it looks at first, because if we just focus in on phenotypes, the physical traits of the offspring, some of these have the same phenotype. For instance, look right here. What color is this plant going to be? Is it going to be short or tall? So purple tall. What's this one going to be? Purple tall in this one. Purple tall in this one. Purple tall in this one. Purple tall, purple tall, purple tall, purple tall. What's this one going to be? Purple short, good. And what's this one going to be? White short, good. And this one's going to be white tall, good. Okay, so anyway, the point is here that there's a lot of purple talls. They don't necessarily have the same phenotype, like this, genotype. This genotype is different from this genotype, but they're both going to be purple talls. So as far as phenotypes are concerned, that's relatively simple. And here's what it is. If you look at those squares and you actually kept track of all the phenotypes you would get, of the 16 possibilities, 9 out of the 16 show dominant for the A trait and dominant for the B trait, purple tall. 3 out of the 16 will show dominant for one trait and recessive for the second trait, so purple white. 3 out of the 16 will show recessive for the first trait and dominant for the second. I should have said purple short. White tall and white short. 1 out of 16 will be recessive, white short in this case. The genotypes are a little more complicated. Here they are, 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1, through all those various genotypes. There we go. But the phenotypes are definitely a little bit simpler to consider. Notice you only get 1 out of 16 chance of having recessive for both traits. That's an interesting thing to remember. There we go. The 9 to 3, the famous 9 to 3 to 3 to 1 ratio. And Mendel actually did this cross, and here's the actual numbers that he got. And again, they don't fall exactly 9 to 3 to 3 to 1, but they're darn close to it, which is what we'd expect for things that are based on a certain element of random chance. All right. Well, let's do this one. Well, let's skip this. We'll do this in the laboratory, but I'll just show you the results here. If I said do this cross, and if you work out the numbers, it would come out to a 3 to 3 to 1 to 1. All right, and I'm going to skip this for the laboratory also. And that. There we go. Okay, so Mendel worked out these laws of genetics in the mid-1800s, and he published his results in a scientific journal. And kind of amazingly, no one paid any attention to them. No one had known up to that point how genetics worked. And then Mendel says, "I've solved it," and publishes it. And everybody just ignored his article. And people are not sure exactly why they ignored it. He published it in kind of an obscure journal. And furthermore, people who did read it probably just thought, well, this is something that applies to plants, but certainly we're very different from plants, so it can't apply to human beings. But they didn't appreciate that what he discovered applied to all living organisms, or at least all sexually reproducing living organisms, including human beings. And so I want to shift now and talk about some dominant recessive traits as far as human beings are concerned. And maybe what I'll do is I'll have two people here, dominant guy and recessive guy over here. One trait that's dominant, easily seen as dominant recessive, are eye color. Brown colored irises in your eyes come from a dominant allele, and blue colored irises in your eyes come from a recessive allele. What the allele is actually for is making melanin, which is a brown or black pigment in your irises. And so if you have a functional copy of that gene, if you have a dominant version allele, you get melanin in your eyes and then brown or black. But if you don't have a dominant copy of that allele, if you have the recessive alleles, you come up with